फिर इसमें आता है एनिमल हस्बेंड्री आता है। अब स्ट्रेटेजी फॉर द एनहांसमेंट इन द फूड प्रोडक्शन इसमें एनिमल हस्बेंड्री है, प्लांट ब्रीडिंग है, सिंगल सेल प्रोटीन है और टिश्यू कल्चर है। तो इनसर्ट ये लिखता है कि विथ ever increasing population of the world, enhancement of the food production is a major necessity. Biological principles as applied to the human animal husbandry and the plant breeding have a major role in our efforts to increase food production. Several new techniques like the embryo transfer technology and the tissue culture techniques are going to play a pivotal role in the further enhancing food production. Animal husbandry है तो animal husbandry is the agriculture practice of the breeding and raising livestock. As such, it is a vital skill for the farmers and is as much science as it is art. Animal husbandry deals with the care and the breeding of the livestock like the buffaloes, cow, pig, sausage, cattle, sheep, camel, goats, etc. that are useful to the humans. Extended, it includes the poultry farming and the fisheries. Fisheries include the rearing, catching, selling, etc. of the fish, mollusca, and the crustaceans, prawn, crabs, etc. since time immemorial. Animals like bees, silkworm, prawn, crabs, fishes, birds, pigs, cattle, sheep, and camels have been widely used by the humans for products like the milk, eggs, meat, wool, silk, honey, etc. It is estimated that more than 70% of the world livestock population is in India and China. However, it is surprising to note that the contribution to the world farm produce is only 25%. That means the productivity per unit is very low. Hence, in addition to the conventional practice of the animal breeding and care, newer technology have also applied to achieve the improvement in the quality and the productivity. फिर इसके बाद आता है मैनेजमेंट एंड ऑफ द फार्म्स एंड द फार्म एनिमल्स. The professional approach to what have been traditional practices of the farm management gives the much needed boost to our food production. Let us discuss some of the management procedure and employed in the various animal farm system. Dairy farm management तो कहता है कि डेयरिंग इज द मैनेजमेंट ऑफ द एनिमल्स फॉर द मिल्क एंड इट्स प्रोडक्ट्स फॉर द ह्यूमन कंजप्शन. कैन यू लिस्ट द एनिमल्स दैट यू वुड expect to find in a dairy? What are the different kinds of the products that can be made with the milk from a dairy farm? In the dairy farm management, we deal with the processes and the system that increase the yield and improve the quality of the milk. Milk yield is a primary dependent on the quality of the breeds in the farm. Selection of the good milk breeds having high yielding potential under the climatic conditions of the area combined with the resistance to the disease is very important. For the yield potential to be realized that the cattle have to be well looked after, they have to be house fed well, should have the adequate water and be maintained disease free. The feeding of the cattle should be carried out in a scientific manner with the special emphasis on the quality and the quantity of the fodder. Besides, stringent cleanliness and the hygiene both of the cattle and their handlers are of the paramount importance while milking, storage and transport of the milk and its product. Nowadays, of course, much of these processes have become mechanized, which reduces the chance of the direct contact of the produce with the handler. Ensuring these stringent measures would of course require regular inspection with the proper record keeping. It would also help to identify and rectify the problems as early as possible. Regular visits by the veterinary doctor would be mandatory. You would probably find it interesting if you were to prepare a questionnaire on diverse aspects of the dairy keeping and then follow it up with a visit of the dairy farm in your locality and seek answer to the question. फिर आते हैं पोल्ट्री फार्म मैनेजमेंट. Poultry is the class of the domesticated false bird used for the food or their eggs. They typically include chickens and the ducks and sometimes turkey and the geese. The word poultry is often used to refer to the meat of only these birds, but in the more general sense, it may be referred to the meat of the other birds too. As in a dairy farming, selection of the disease-free and the suitable breeds, proper and the safe farm conditions, proper feed and the water, and the hygiene and the health care are the important components of the poultry farm management. कहता है कि you may have seen TV news or the read newspaper about the reports about the bird flu virus which created a scare in the country and drastically affected eggs and the chicken consumption. Found out more about it and discussed whether the panic reaction was justified. How can we prevent the spread of the flu in the case of the some chicken are infected? इसके बाद फिर आता है एनिमल ब्रीडिंग. तो ब्रीडिंग ऑफ द एनिमल्स इज एन important aspect of the animal husbandry. Animal breeding aims at increasing the yield of the animals and improving the desirable qualities of the produce. So what kind of the character would be breed animals? For would the selection of the character differs with the choice of the animals? What do we understand by the term breed? A group of the animals rated by the descent and similar in the most characters like the general appearance, feature, size, configuration, etc. are said to belong to the breed. Find out the names of the some common breeds of the cattle in the poultry in the farms of your area. When breeding is between animals of the same breed, it is called as the inbreeding, while the cross between the different breed is called as the outbreeding. तो इनब्रीडिंग क्या होता है? इनब्रीडिंग refers to the mating of the more closely related individuals within the same breed for the fourth to fifth generation. The breeding strategy is as follows: superior males and the superior females of the same breed are identified and mated in the pairs. The progeny obtained from the such matings are evaluated, and superior males and the females among them are identified or for their mating. A superior female in the case of the cattle is the cow or the buffalo that produces more milk per lactation. On the other hand, a superior male is the bull, which gives rise to the superior progeny as compared to those of the other males. Try to recollect the homozygous pure lines developed by the Mendel as I discussed in chapter 5. A similar strategy is used for developing pure lines in the cattle as was used in the peach case of the peach. Inbreeding increases the homozygosity, thus inbreeding is necessary if we want to evolve a pure line in any animals. Inbreeding exposes harmful recessive genes that are eliminated by the selection. It also helps in the accumulation of the superior genes and the elimination of the less desirable genes. Therefore, this approach, where there is selection at each step, increases the productivity of the inbred population. However, continued inbreeding, especially close breeding, usually reduces fertility and even productivity. This is called inbreeding depression. Whenever this becomes a problem, selected animals of the breeding population should be mated uh, with the unrelated superior animals of the same breed. This really helps restore fertility and yield. Outbreeding is the breeding of the unrelated animals which may be between the individuals of the same breed but having no common ancestor for the fifth to sixth generation. Outcrossing or between different breeds, crossbreeding or different species, interspecies hybridization. फिर आता है आउट क्रॉसिंग तो दिस इज द प्रैक्टिस ऑफ द मेटिंग ऑफ द एनिमल्स विद द सेम ब्रीड but having no common ancestor on either side of their pedigree up to the fourth to sixth generation. The offspring of such a mating is known as an outcross. It is the best breeding method for the animals that are below average in productivity in the milk production, growth rate in the beef cattle, etc. A single outcross often helps to overcome the inbreeding depression. इसके बाद फिर आता है क्रॉस ब्रीडिंग तो इन दिस मेथड सुपीरियर मेल्स ऑफ वन ब्रीड आर मेटेड विद द सुपीरियर फीमेल्स ऑफ द अनदर ब्रीड क्रॉस ब्रीडिंग अलाउज द डिजायरेबल क्वालिटी ऑफ द टू ब्रीड different breeds to be combined. The progeny hybrid animals may themselves be used for the commercial production. Alternatively, they may be subjected to one form of the breeding and selection to develop a new stable breeds that may be superior to the existing breeds. Many new animal breeds have been developed by this approach. Hiffardale is a new breed of the sheep developed in the Punjab by crossing Bikaner ewes and the Merino rams. इसके बाद फिर आता है interspecies hybridization. In this method, male and the female animals of the two different related species are mated. In some cases, the progeny may combine the desirable feature of both the parents and may be of the considerable economic value. Example, the mule. Do you know what cross leads to the cross production of the mule? इसके बाद फिर आता है controlled breeding experiment. Our controlled breeding experiments were carried out using the artificial insemination. The semen is collected from the male that is chosen as the parent and injected into the reproductive tract of the selected female by the breeder. The semen may be used immediately or can be frozen or and used at a later date. It can also be transported in a frozen form to where the female is housed. In this way, desirable mating are carried. Artificial insemination helps to overcome several problems of the normal mating. Can we discuss and list some of them? Often, the success rate of the crossing mature male and the female animals is fairly low enough, though artificial insemination is carried out to improve chances of the successful production of the hybrids. Other means are also used. Multiple ovulation embryo transfer technology is one such a program for the herd improvement. In this method, a cow is administered hormones with the FSH-like activity to induce the follicular maturation and superior ovulation instead of the one egg which they normally yield per cycle. They produce 6 to 8 eggs. The animal is either mated with an elite bull or the artificial inseminated. The fertilized eggs at 8 to 32 cell are recovered non-surgically and transferred into surrogate mothers. The genetic mother is available for another round of the superovulation. This technology has been demonstrated for the cattle, sheep, rabbit, buffaloes, mares, etc. High-yielding milk varieties of the females breed and high-quality lean meat with the less lipid milk-yielding bulls have been bred successfully to increase the herd size in a short time. ये म्यूल का है जो पिक्चर दिए हुए है. इसके बाद फिर ये दिए हुए है जर्सी और ये है लेगहॉर्न से इम्प्रूव्ड वेराइटी ऑफ द कैटल एंड द चिकन है. इसके बाद फिर आता है बी कीपिंग. तो beekeeping or the apiculture is the maintenance of the hives of the honeybees for the production of the honey. It has been an age-old cottage industry. Honey is a food of the high nutritive value and also found used in the indigenous system of the medicine. Honeybee also produces bee wax, which finds many uses in the industry, such as in the preparation of the cosmetics and polyphage of the various kinds. The increased demands of the honey has led to the large-scale beekeeping practices. It has become an established income generating industry whether practiced on a small or on a large scale. Beekeeping can be practiced in many areas where there are sufficient bee pasture of some wild shrubs, fruit orchards, and cultivated crops. These are several species of the honeybee which can be reared. Of the most common species is the Apis indica. Beehives can be kept in the one's courtyard, on the veranda of the house, or even on the roof. Beekeeping is not labor intensive. Beekeeping, though relatively easy, does not require some special knowledge, and there are several organizations that teach beekeeping. The following points are important for the successful beekeeping. पहला, knowledge of the nature and the habits of the bees. दूसरा, selection of the suitable location for the keeping the bees. तीसरा, catching and having the swarms group of the bees. चौथा, management of the beehive during the different seasons and handling and collection of the bees and the bees bags. Bees are the pollinators of many our crop species such as the sunflower, brassica, apple, and the pear. Keeping beehives in the crop fields during the following period increase the pollination efficiency and improve the yield, beneficial both from the point of the view of the crop yield and the honey yield. इसके बाद फिर आता है फिशरीज. तो फिशरी इज एन इम्पॉर्टेंट इज एन इंडस्ट्री डिवोटेड टू द कैचिंग प्रोसेसिंग और द सेलिंग ऑफ द फिशेज शेलफिश और द अदर aquatic animals. A large number of our population is dependent on the fish products and other aquatic animals such as the prawns, crabs, lobster, edible oyster, etc. for food. Some of the freshwater fishes which are common include the catla, rohu, common carp. Some of the marine fishes that are eaten include the hilsa, sardines, mackerel, and the pomfrets. Find out what fish are commonly eaten in your area. इसके बाद फिर आता है कि fisheries has an important place in the Indian economy. It provides income and employment to millions of the fishermen and the farmers, particularly in the coastal states. For many, it is only the source of their livelihood. In order to meet the increasing demand on the fisheries, different techniques have been employed to increase the production. For example, though the agriculture and the pisciculture, we have been able to increase the production of the aquatic plants and animals for both freshwater and the marine. Find out the difference between the pisciculture and the agriculture. This has led to the development and the flourishing of the fishery industry, and it has brought a lot of income to the farmers in particular and the country in the general. We now talk about the blue revolution has been implemented along the same line as the green revolution. फिर आता है प्लांट ब्रीडिंग. तो traditional farming can only yield a limited biomass as food for the humans and the animals. Better management practices and increase in the acreage can increase the yield, but only up to a limited extent. Plant breeding also as a technology has helped increase the yields to very large extent. Who in India has not heard of the Green Revolution, which was responsible for our country to not merely meet the national requirement in the food production, but also helped us to even export. Green Revolution was dependent to a large extent on the plant breeding techniques for the development of the high yielding and the disease-resistant variety of the wheat, rice, and the maize, etc. Prata Bhati plant breeding. So plant breeding is the purposeful manipulation of the plant species in order to create a desired plant types that are better suited for the cultivation, give the better yields, and are disease resistant. Conventional plant breeding has been practiced for the thousands of the years since the beginning of the human civilization. Recorded evidence of the plant breeding dates back to 11,000 years ago. Many present day crops are the result of the domestication in the ancient times. Today, all our major food crops are derived from the domesticated varieties. Classical plant breeding involves crossing or the hybridization of the pure lands, followed by the artificial selection to produce the plant with the desirable traits of the higher yield, nutrition, and resistance to the disease. With the advancement in the genetics, molecular biology, and the tissue culture, plant breeding is now increasingly being carried out using the molecular genetic tools. If we are to list the traits or the characters that the breeders have tried to incorporate into the crop plants, the first we would list would increase the crop yield and the improved quality, increase tolerance to environmental stress, salinity, extreme temperature, and the drought resistant to the pathogen, virus, fungi, and the bacteria, and increase tolerance to the insect pest to be our list too. फिर आता है कि plant breeding program are carried out in a systematic way worldwide in government institutions and commercial companies. The main steps in the breeding a new variety of the genetic crop are collection of the variability. Genetic variability is the root of any breeding program. In many crops, pre-existing genetic variability is available from the wild relatives of the crop. Collection and the preservation of all the different wild varieties of species and the relatives of the cultivated species, followed by their evolution for the characteristic, is a prerequisite for the effective exploitation of the natural genes available in the population. The entire collection of the plant feed having all the diverse allele for the genes in a given crop is called a germplasm collection. Prior to evolution and the selection of the parents, the germplasm is evaluated so to identify the plants with the desirable combination of the characters. The selected parents were multiplied anew to in the process of the hybridization. Pure lines were created wherever deifiable and possible. इसके बाद आता है क्रॉस हाइब्रिडाइजेशन among the selected parents. The desired characters have been very often to be combined from the two different parents. For example, high protein quality of one parent may need to be combined with the DA resistant from the another parent. This is possible by the cross hybridizing the two parents to produce the hybrid that genetically combine the desired character in one plant. This is a very time consuming and tedious process since the pollen grains from the dual parents chosen as the male parent have to be collected and placed on the stigma of the flower selected as the female parent. In chapter 2, details how to make the crosswise have been described. Also, not necessary that they have to combine to diable character. Usually, only one in the few hundred to a thousand cross for the diable combination. इसके बाद फिर आता है सिलेक्शन एंड टेस्टिंग ऑफ द सुपीरियर रिकॉम्बिनेंट्स. दिस स्टेप consists of the selecting among the progeny of the hybrids those plants that have the desired character combination. The selection process is crucial to the success of the breeding objectives and requires careful scientific evaluation of the progeny. This step yields plants that are superior to the both of the parents. Very often, more than one superior progeny plant may become available. These are self-pollinated for the several generations till they reach a state of the uniformity, homozygosity, so that the characters will not segregate in the progeny. फिर आता है टेस्टिंग रिलीज एंड द कमर्शलाइजेशन ऑफ द कल्टीवार्स. दी newly selected lands are evaluated for their yield and the other agronomic traits of the quality. DJ resistant. This evaluation is done by the growing trees in the research fields, recording their performance under the ideal fertilizer application, irrigation, and the other crop management practices. The evolution in the research field is followed by testing the materials in the farmer's field or at least 3 growing seasons at several locations in the country, representing all the agroclimatic zones where the crop is usually sown. The material evaluated in the comparison to the best available local crop cultivar are check of the reference cultivar. India is mainly an agriculture country. Agriculture accounts for approximately 33% of the India GDP and employ nearly 52% of the population. After Indian independence, one of the main challenges facing the country by that of producing enough food for the increasing population, as only limited land is fit for the cultivation. India has to strive to increase the yields per unit area from existing farmland. The development of the several high-yielding varieties of the wheat and the rice in the mid-1950s, as a result of the various plant breeding techniques, led to the dramatic increase in the food production in our country. This phase is often referred to as the Green Revolution. Figure 9.3 represents some Indian hybrid crops of the high-yielding varieties. तो ये दिया हुआ है सम इंडियन हाइब्रिड क्रॉप, ये मेज का दिया हुआ है, व्हीट का दिया हुआ है और गार्डन पीच का दिया हुआ है. P1542 जो है गार्डन पीच का आता है. सोनालिका जो है व्हीट का है और मेज का दिया हुआ है. इसके बाद फिर आता है व्हीट एंड द राइस में देख लेते हैं. तो during the 1950 to 2000, wheat production increased from the 11 million tons to 75 million tons, while the rice production went up from the 35 million tons to 89.5 million tons. Despite due to the development of the semi-dwarf variety of the wheat and the rice, Nobel laureate Norman Borlaug at International Center for the Wheat and the Maize Improvement in the Mexico developed semi-dwarf wheat. In 1953, several varieties such as the Sonalika and the Kalyansona, which were high-yielding and disease-resistant, were introduced all over the wheat-growing belt of the India. Semi-dwarf rice varieties were developed from the IR8, developed at International Rice Research Institute (IRRI), and the Taichung native one from the Taiwan. The derivatives were introduced in the 1956. Later, a better yielding semi-dwarf variety Jaya and the Ratna were developed in the India. इसके बाद फिर आता है शुगर केन. तो सैक्रम बारबेरी was originally grown in the North India but had poor sugar content and the yield. Tropical canes grown in the South India. Saccharum officinarum had a thicker stem and higher sugar content but did not grow well in the North India. These two species were successfully crossed to get the sugarcane varieties, combining the desirable quality of the high yield, thick stem, high sugar, and the ability to grow in the sugarcane areas of the North India. फिर आता है millets. तो hybrid maize, ज्वार and the bajra have been successfully developed in the India. Hybrid breed have led to the development of the several high-yielding varieties resistant to the batter of Treska. फिर आता है plant breeding for the disease resistant. तो कहता है कि a wide range of the fungal, bacterial and the viral pathogens affect the yield of the cultivated crop species, especially in a tropical climate. Crop losses can often be significant, up to 20 to 30%, or sometimes even total. In this situation, breeding and the development of the cultivars resistant to the disease enhances food production. This also helps reduce the dependence on the use of the fungicide and the bactericides. Resistance of the host plant is the ability to prevent the pathogen from causing disease and determined by the genetic constitution of the host plant. Before breeding is undertaken, it is important to know about the causative organism and the mode of the transmission. Some of the diseases caused by the fungi are the rust, example brown rust of the wheat, red rot of the sugarcane, and the late blight of the potato. By the bacteria, black rot of the crucifers, by the virus to acomosaic turnip virus. फिर आता है मेथड ऑफ ब्रीडिंग फॉर द डिजीज और रेसिस्टेंट तो ब्रीडिंग इज कैरिड out by the conventional breeding techniques described earlier or by the mutation breeding. The conventional method of breeding for the disease resistant is that of the hybridization and the selection. Its steps are essentially identical to those of the breeding for any other agronomic characters such as the high yield. The steps, very sequential steps are screening germplasm for the resistance force, hybridization of the selected parent, selection and the evolution of the hybrids and testing and the release of the new varieties. Some crop varieties held by bred by the hybridization and the selection for the DJ resistant, the fungi and bile DJ are related. तो भीट है जो हिमगिरी कहता है लीफ एंड स्टाइप एंड हिल बंट कहता है ब्रासिका पूफा फन में भाइट रफ्ट कहता है करण राय में इसके बाद कॉलीफ्लावर पूफा फुबरा पूफा नोबल के वन है जो ब्लैक रॉट एंड द कल ब्लैक रॉट कहता है फिर कॉपी पुफा कॉमन बैक्टीरिया ब्लाइट कहता है चिल्ली पुफा फदाबहा चिल्ली मोफेक बार ऑफ टोबैको मोफेक बार ऑफ लीफ कल कहता है तो आता है कि कन्वेंशनल conventional breeding is often constrained by the ability of the limited number of the DA-resistant genes that are present and identified in the various crop varieties or wild relatives, inducing mutations in the plant through the diverse means and then screening the material plant for the resistance sometimes lead to the derived genes being identified. Plants being their rival characters can then be either multiplied directly or can be used in the breeding. Other breeding methods that are used are the selection amongst the pharmacogonal variant and the genetic engineering. इसके बाद फिर आता है म्यूटेशन तो म्यूटेशन इज द प्रोसेस बाय विच द जेनेटिक वेरिएशंस आर क्रिएटेड थ्रू द चेंजेस इन द बेस सीक्वेंस विद इन द जीन्स resulting in the creation of a new character or the trait not found in the parental type. It is possible to induce mutation artificially through the use of the chemical for the radiation like the gamma radiation and the selecting and using the plants that have the dual characters as a source of in the breeding. This process is called the mutation breeding in the mung bean resistant to the yellow mosaic virus and the powdery mildew were introduced by the mutations. इसके बाद फिर आता है कि several wild relatives for the different cultivated species of the plants have been shown to certain resistant character but have very low yield. Hence, there is a need to introduce the resistant genes into the high-yielding cultivated variety resistant to low mosaic virus in the building. Abel muscus esculenthus by transferred from a wild species and resulted in a new variety, Abel esculenthus called the Pravani Kranti. इसके बाद फिर आता है कि all the above examples include the sources of the resistance genes that are in the same crop species which highlight to the bred for the DA resistance or in a related wild species. Transfer of the resistant genes is achieved by the sexual hybridization between the target and the source plant followed by the selection. इसके बाद फिर आता है plant breeding for the developing resistance to the insect pest. Another major cause for the large-scale destruction of the crop plant and the crop produce is insect and the pest infestation. Insect resistance in the host crop plant may be due to the morphological, biochemical, or the physiological characteristic. Hairy leaves from the soil plants are associated with the resistance to the insect pest, example resistant to the aphids in the cotton, and cereal leaf beetle in the wheat. In the wheat, solid stems lead to a non-preference by the stem softfly and the smooth-lipped and the nectarless cotton variety do not attract bollworms. High aspartic acid, low nitrogen and the sugar contained in the maize lead to the resistance to the maize stem borers. Breeding methods for the insect pest resistant involve the same step as those for any other economic trait such as the high yield or the quality and are discussed earlier. Sources of the resistant genes may be cultivated varieties, germplasm collection of the crop or the wild relatives. Some related crop variety is bred by the hybridization and the selection for the insect persistence are given in table 9.4. Brassica, Pufa Gaurav aphids कहता है, flat bean Pufa fem 2, Japhids aphids and the Pufa fem 3, fruit borer कहता है, फिर ओकरा भिंडी Pufa स्वानी fruit and the fruit borer होता है Pufa A 4 होता है इसका. इसके बाद फिर आता है प्लांट ब्रीडिंग फॉर द इम्प्रूव्ड क्वालिटी तो कहता है कि मोर देन 840 मिलियन पीपल इन द वर्ल्ड डू नॉट हैव द एडुकेट फूड टू मीट देयर daily life and nutritional requirements. A far greater number, 3 billion people, suffer from the macronutrient, protein and the vitamin deficiency or the hidden hunger, 3 billion, because they cannot afford to buy enough fruit, vegetables, legumes, fish and the meat. Diets lacking essential macronutrients, particularly iron and vitamin A, iodine and zinc, increase the risk for the disease, reduce lifespan and reduce the mental abilities. इसके बाद फिर आता है बायोफोर्टिफिकेशन तो Breeding crops with the high levels of the vitamins and the minerals or the higher proteins and the healthier fats with the most practical means to improve the public health. Breeding for the improved nutritional quality is taken with the objective for the improving protein content and the quality, oil content and the quality, vitamin content and the micronutrient and the mineral content. In the maize, in the tooth maize habits that have twice the amount of the amino acids in the lysine in the tryptophan compared to the existing maize habits were developed. Wheat variety Atlas 56 having a high protein content has been used as a donor for the improving cultivated wheat. It has been possible to develop an iron fortified rice variety containing other over 5 times as much iron as commonly consumed varieties. The Indian Agriculture Research Institute, New Delhi, has also released several vegetable crops that are rich in the vitamins and the minerals. Vitamin A enriched carrot, spinach, pumpkin, vitamin C enriched bitter gourd, bathua, mustard tomato, iron and calcium enriched spinach and the bathua, and the protein enriched beans, broad, level of French and the garden peas. फिर आता है सिंगल सेल प्रोटीन एससीपी तो कहता है कि conventional agriculture production of the cereals, pulses, vegetables, fruits, etc. may not be able to meet the demand of the food at the rate at which the human and the animal production is increasing. The shift from the grain to the meat diet also creates more demand for the cereals. It takes 10 kg of the grain to produce 1 kg of the meat by the annual farming. Can you explain this statement in the light of the knowledge of the food chains? More than 25% of the human population is suffering from the hunger and malnutrition. One of the alternate source of the protein for the animals and the human nutrition is the single cell protein. Microbes are being grown on an industrial scale as a source of the food protein. Green algae like the spirulina can be grown easily on the materials like the wastewater from the potato processing plants containing starch, straw, molasses, animal manure, or even sewage to produce a large quantity and conserve a food rich in the protein, animals, fats, carbohydrates, and the vitamins. Incidentally, such utilization also reduces environmental pollution. Certain bacterial species like the Methylophilus methyllotropus, because of its high rate of the biomass production and growth, can be expected to produce 25 tons of the protein. The fact that edible mushrooms are eaten by many people and large-scale mushroom culture is going industry make it believable that microscopic fungi too would become acceptable as a food. इसके बाद फिर आता है टिश्यू कल्चर तो as a traditional breeding technique failed to keep pace with the demand and to provide a sufficiently fast and efficient system for the crop improvement. Another technology called the tissue culture got developed. What the tissue culture mean? It was learned by the scientists in the 1950s that whole plant could be regenerated from the explant, that means any part of the plant taken out and grown in a test tube under sterile condition in a special nutrient media. This capacity to regenerate a whole plant from any cell explant is called a totipotency. You will learn how to accomplish this in the higher classes. It is important to stress that a nutrient medium must provide a carbon source such as the sucrose and also inorganic salts, vitamins, amino acids, and the growth regulators like the oxygen, cytokinins, etc. By application of this method, it is possible to achieve propagation of a large number of the plants in a very short duration. This method of the producing thousands of the plants for the tissue culture is called the micropropagation. Each of these plants will genetically identical to the original plant from which they were grown, that means soma clones. Many important food plants like the tomato, banana, apple, etc. have been produced on a commercial scale using this method. Try to be at a tissue culture laboratory with your teacher to better understand and appreciate the process. Another important application of the method is the recovery of the healthy plants from the disease plant. Even if the plant is infected with a virus, the meristem, apical and axillary is free of the virus, hence one can remove the meristem and grow it into in vitro to obtain the virus-free plant. Scientists have succeeded in culturing meristem of the banana, sugarcane, potato, etc. Scientists have evolved even isolated single cell from the plants, and after digesting their cell walls have been able to isolate naked protoplasts surrounded by the plasma membranes. Isolated protoplasts from two different varieties of the plants, each having a desirable character, can be fused to get hybrid protoplasts which can be further grown to form a new plant. These habits are called somatic habits, which the process is called somatic hybridization. Imagine a situation when a protoplast of the tomato is fused with that of the potato. When they are grown to form new hybrid plant combining tomato and the potato characteristic, well, this had been achieved, resulting in the formation of the tomato. Unfortunately, this plant did not have all the desired combination of the characteristic for its commercial utilization. इसके बाद फिर आता है इसका समरी दिया हुआ है कि Animal husbandry is the practice of taking care and breeding domestic animals by applying scientific principles. The ever increasing demand of the food from the animals and the animal products, both in the terms of the quality and the quantity, have been met by the good animal husbandry practices. These practices include the management of the farm and the farm animals and the animal breeding. In view of the high nutritive value of the honey and its medicinal importance, there have been a remarkable growth in the practice of the beekeeping or the apiculture. Fishery and other flourishing industry meeting the ever-increasing demand for the fish production and other aquatic foods. Plant breeding may be used to create the varieties which are resistant to the pathogen and to the insect pest. This increase the yield of the food. This method has also been used to increase the protein content of the plant foods, thereby enhance the quality of the food. In India, several varieties of the different crop plants have been produced. All these measures enhance the production of the food. Techniques of the tissue culture and the somatic hybridization offer vast potential and manipulation of the plants in vitro to produce the new varieties.